my next couple of final slides. So this is a slide that I would like to that I basically got the idea from one of our collaborators, Dr. Rebecca Bott, in the Donald Danforth Center. We are collaborating with them in different projects in US. And her idea, I like this because her idea is that she is using it for plant diseases, but I modified it and tried to use it for plant-virus interaction research, where I find that we need to create, we need to have foundational knowledge. So which means we need to have basic research undergoing, and then that should be used in the translational research. And this circle should continuously keep going on to move the area of plant-virus interaction research forward. So with this, I will be ending my slide, but before that, I would like to acknowledge the lab where I did this work and my present lab. So I did my PhD in Dr. Ellen Sanderson's lab at the University of British Columbia. It was fun time over there. We used to go for a lot of skiing or snowshoeing, and this is one such event. I really liked it. And then I would like to thank my present PI, Professor Stephen E. Jacobson. Here I am also into several projects where I am developing CRISPR-based epigenome editing tools, as well as I'm developing plant viral delivery tools. This is fun time here. And finally, I would like to thank my undergraduate students. They are, without them, I don't think my research life would be so livable, because they provide energy to me as well. So I'm mentoring a lot of undergraduate students here, and this is mostly the whole list of undergraduate students whom I have mentored, and they have, some of them have moved ahead. Some of them are doctors, some of them are researchers now. It's amazing to have them. And with this, finally, I would like to say thank you to all the audience I have here for listening to my talk, and also have the patience to listen to me. And I like this slide at the very end, and this is mostly to the young researchers or young undergraduate students or postgraduate students who are looking at this slide, is that most of the days when we are working in the lab, we ask the question, what is the opposite of eureka? And so most of the times experiments fail. It's like 99% of times. And only 1% of time, you really get a chance to shout out loud and say eureka. So thank you. I'm ready to take questions now.
Thank you. Now we have some of questions, like 13 questions. You can choose a few of them from question answer box and can answer them. And I have one question or two questions from YouTube also. So please go for the questions from the question answer box.
So there is a question, I'm not sure what the question is, but this is from Ganesh Satarkar, asking that the difference between RNA and DNA viruses. I would say this is a very basic question. That is, the viral genome is different between the RNA and the DNA viruses, and therefore the multiplication cycle of the RNA and then the DNA between the RNA virus and the DNA viruses are different. So I'm not going into the details of this question. The potato which, so there is this interesting question that the potato which gets infected with potato virus Y can harm humans, can cause harm to humans. This is from Shiva Maurya. I don't think there are any reports of plant viruses infecting humans. So obviously the potato virus Y won't infect the humans. Any known which Gemini virus which we can target using CRISPR? Okay, there is this another question. Any known host protein except pCNA which interact with Gemini virus protein, which we can target using CRISPR-Cas technology? At this point, nothing is coming to my mind. So I'm sorry. I think if you Google, if you look at the literature, you might find some. For example, with the RNA viruses, I will go back to RNA viruses because that's where I fear that's what I have worked with a lot, and also currently the projects that I am involved in. One of them actually is with one particular RNA virus. And for RNA viruses, some of the RNA viruses, they do use certain kind of elongation factors like EIF iso4E. They are being targeted. And I think here in the first CRISPR-Cas talk, that question was addressed. The next question we have is from Shiva Maurya. Is symptoms recovery is also possible in susceptible plants? That's actually a very good question. So I am not sure about it, but you know, I had done certain experiments where I would take the plants that were highly infected, that got highly infected at 21 degrees centigrade, and then I will move them to higher temperatures. Sometimes they would recover, but it also depended on the fact that how far the viruses were able to spread. The next question is from Ravindra Sadbhai. Will temperature silence the infection of virus? Now, I don't think that it's temperature that silences the infection of the virus. It's basically the mechanisms, the molecular mechanisms, like whether it's the resistance pathways, like the R gene mediated pathways or RNA silencing mechanism, that gets cleared away. Yeah, that actually clears the viruses. So suppose in this case, in my case, when I was doing the research, at higher temperatures, you would get symptom recovery because you have a higher, stronger RNA silencing taking place at higher temperatures. The next question is, does other environmental factors like photoperiod have any effect on translation repression in case of tomato ring spot virus? Now, we never checked the effect of photoperiods. Never checked the photoperiods with translation repression. But I would say that I was also, while I was in Canada, right after my PhD, I was in a lab for around 9 months in Dr. Deanne Roshan's lab at the University of British Columbia. And one of the projects was actually to study the effect of light on virus infection. And light obviously affects the virus infection, because with light, it's like when you're talking about light, you have to think about chloroplast, and chloroplasts are involved in several several plant defense responses. So you have to keep that in mind, that that there can be effect on the chloroplast, and that can have an effect on the virus infection. This is okay. This is a question from Dr. Akhilesh K. Kusawa. He's a postdoctoral fellow at IISc Bangalore, and his question is, do you think that abiotic stress priming, for example, heat stress priming, will enhance plant resistance or plant virus multiplication or virus movement or code protein synthesis? I would say that this is a good question. There obviously is a possibility, because once you are providing a particular kind of stress to a plant, then you are actually priming the plant for that particular kind of stress. So you're basically reprogramming the gene expression. of that plant. And there can be some genes that are also involved in controlling the other pathways. And so that might have an effect on plant virus multiplication or virus movement. In line with this, I would like to add that there are examples in plants. For example, for example, with Arabidopsis actually, if you infect Arabidopsis with cucumber mosaic virus, these plants were found to be drought tolerant. So they would sustain drought for a longer period of time. So obviously there can be an effect. Viral RNA in presence in recovered leaves, it interferes with any metabolic activities of them. Then we have another question from Isha Madan, that viral RNA in presence in recovered leaves, does it interfere with any metabolic activities of plant? Honestly, we didn't look at other metabolic activities of the plant, but what our concept was that the virus in the recovered leaves is basically is not getting translated. And we were thinking that some of our other experiments that I didn't talk about here, that the viral protein was actually, if there is viral protein present, then that was inducing certain difference response genes like PR1A. So now if you have low levels of viral proteins, those genes like PR1A were not getting activated. And we obviously have shown this in our work. I just didn't talk about it here. The next question is from Abhishek Samal. Does temperature affect all the virus across plants, or it's only specific for TORSV? No, temperature affects, the effect of temperature, I would say, is mostly on all the viruses across plants. Yeah, it's not only TORSV. There are several other examples where temperature was shown to have an effect on the virus multiplication or viral infection. Is there any relation between mutation of plant RNA virus and inoculation of resistant plant virus? I am not sure about this question. And inoculation of resistant plant virus. Mutations of RNA plant virus. I think what is meant here is that since RNA viruses are prone to mutation, is there any relationship between that? And at this point, I'm not sure about the question, so I can obviously try to answer it later if I get the question in a more explicit form. Oh. Is there any mechanism of PR1 expression and signaling involving plant virus? How is it different? Is there any mechanism of PR1A and SA induced signaling involved? How it is different from bacteria? Now, in this case, there is obviously mechanisms for PR1A expression and SA induced signaling in the plant virus interaction. Then, the difference here is there's not, I won't say that there is a huge difference, but obviously you have to think the way a virus infects and the bacteria infects. So a virus doesn't get into the cell, right? So in case of in case of bacteria, the bacteria doesn't get into the cell. A bacteria actually stays outside the plant cell, and then it tries to manipulate the plant cell to get nutrition from the plant cell. So there are receptors on the plant cell that recognize certain receptors, or not receptors, but certain proteins like the flagellins on bacteria, and that triggers like PTI or PAM triggered immunity or effector triggered immunity for the bacteria. But in case of viruses, they are directly injected within the plant cell, and that's a huge difference. So that is obviously one of the major difference, I would say. Please let me know about the interaction between Viroid and plants. Okay, so there's one question. Please let me know about the interaction between viroid and plant. Now, this is a great question. And obviously now viroids are basically similar to viruses, but they don't encode for their own protein. And that's, if I started, that's another huge field. Like one of my friend is working on it, and that's his career for the last 15-18 years. That's what he's working. So if I go into details about all of this, that will be that will be a long time to talk about. So I think that's all. I would before I finish, I would like to, this is from Shilpa N, and I would like to tell her, you will find lots of literature on viroid and plant interaction. So kindly go and look